Today, we'll be going over viability assays, which are a critical part of a lot of cell biology research, including those completed at the Bloom lab, such as the chromosome breakage research. So, first, we got to define what is cell viability. In its most basic definition, it's the proportion of living cells in a population. This doesn't necessarily indicate the level of health. Since it's based off the principles of cell division, viable cells are considered those that can divide and proliferate, and dead or arrested cells, which are considered unviable, are the ones that do not divide or proliferate. So, hypothetically, this would mean that cancer cells are considered a viable cell, even though we as biologists understand that cancer cells are not a good thing. Okay, so what's a viability assay then? Well, we define what cell viability is. Now we want a way to measure this quantitatively since it is a proportion or a percentage. So viability assays is a broad category of assays, and they are designed to determine cell viability using different means. For example, there's crystal violet proliferation assays, which are more likely to determine the proliferation of cells. And this uses crystal violet, which is just a violet dye, as you can see in the top right corner. On the other hand, trypan blue exclusion assays use trypan blue, which is indicated there. And they actually measure more so the amount of dead cells versus the amount of living cells. So what are some example procedures? Well, we can start with the yeast spot assay that was used at the Bloom lab. For a bit of context, YPJ is yeast extract, peptone, and glycerol. YPD is the same thing, but instead of glycerol, dextrose is used, which is a type of glucose. So in this procedure, the cell strain of interest is grown on a YPJ medium, and then it's continually diluted in sterile water at different concentrations. So these are all now spread onto YPD and YPG plates, which are then incubated at 24 degrees Celsius. And then after this incubation, the individual colonies are counted for both plates at different concentrations. And the percent viability is calculated using the number of YPD plates or YPD colonies and dividing it by the YPG colonies. So you can see here, this is an example of the spot assay, but for a researcher, this is a real pain since you don't want to be counting all of those. So there's more electronic and computational means of doing this without manual counting. For example, the MTT assay, in this procedure, you can pipette cells of interest. It doesn't necessarily have to be yeast cells into wells and then incubate it at 37 degrees Celsius for 24 hours. You then add MTT, which is a salt dye, to the wells, incubate it again for 4 hours, and then you add a solubilization solution, which just increases the solubility of the plate. And then incubate it one more time overnight, and then you can use an ELISA reader to quantify the microplate color results. If you don't know what an ELISA reader is, it can use absorbance of the wells to determine the concentration of cells or to make logarithmic graphs of the absorbance. In this case, as we go left to right, we can see that the wells become gradually darker. And if you were using an ELISA reader, at the very right side, you'd probably have the greatest result for absorbance because you may remember from your physical science class or your chemistry class that darker usually indicates higher absorbance. And why exactly should we measure cell viability? Well, it provides a lot of interesting insights into biological concepts. For example, there is a cell viability assay regarding ATP, and if we can tell that a certain cell is not viable in a certain condition, we could hypothetically say that using the ATP assay, that cell is not generating enough or any ATP. Additionally, we could talk about cancer development on the opposite end of the spectrum, because if a cell is proliferating at an insane amount in a situation where it shouldn't be proliferating, but it still is, then we could say that there's a cancer possibly being developed. Finally, it could also be used in medical testing. A lot of diseases such as muscular dystrophy and myotonic dystrophy have degenerative cells. And in the case of muscle cells, if we were able to determine that a lot of those cells or the cell viability was decreasing rapidly, then we could say that we could provide treatment before it becomes a more symptomatic and complicated situation. Of course, if you want more information, feel free to use any of these sites and have a wonderful day.